Hi everyone, my name's Andre and welcome to Med School EU. Today we're going to do a lesson on the eye anatomy and physiology. So we're nearing the end of the anatomy and physiology lectures, and this is getting exciting because we're also nearing the end of the biology section altogether. So for today we are going to be discussing the anatomy of the eye. And then we're going to talk about how the eye functions, how light gets through, and how we perceive it within our brain. And of course, the first thing we got to do is begin with the eye anatomy. So we're going to discuss all of the little structures associated with the eye and what they mean and generally what they do. And so before we do that, we're going to have to label the structures so we can understand how the eye is able to function. Now, the first thing we've got here on the most outer structure is called the sclera. Now, if we're going more innerwards towards the center of the eye, we've got this structure called the choroid. So it's called the choroid layer. And finally, the innermost structure of the back of the eye is called the retina. And this is an important structure that contains rods and cones, which are cells that are able to sense the light that's coming in. They're able to take in the information from the light that's coming in and be able to transfer it over to the optic nerve. As you can see here, the retina, all of that has rods and cones, and it collects information, and it all ends up in the optic nerve. Optic nerve is the one that goes all the way to the brain. It goes through the optic chiasma, which we will discuss, and then our brain is able to perceive light and is able to perceive images and construct images within our brain the way we see them and the way the light hits the rods and the cones that are on the retina. Now, this interesting part right here, this little dip on the retina, is called the focal point. The focal point is basically just a very condensed area of focus. So this is primarily where all of the rods and cones are located. Not all of them, but there's just a big cluster of rods and cones located at the focal point of the retina. So going in closer to the front of the eye, we've got conjunctiva, and this is kind of the watery layer that's in front of the eye. It's a covering, it's a protective layer in front of the lens and in front of the other structures. So the conjunctiva and the cornea, which is the inner part of this layer, so the cornea, these structures do not get vascular supply. So capillaries and the blood vessels do not reach the cornea and the conjunctiva structures. So this is actually was a question on the 2021 IMAT exam, and this is kind of the knowledge I guess they expected you to know. So this is something important to remember, that the cornea and the conjunctiva would not have vascular supply. When we're talking about the color of our eyes, that is denoted by the iris. As you can see, the iris comes as the structure that is on the sides of the lens. It's covering the sides of the lens, and that is the structure that would give our eyes the color that we have. Now, of course, this structure is called the lens. That is what reflects light that is going to be concentrated onto the back of the eye, and that will be perceived by the rods and the cones. And finally, the last structure I gotta mention here is called the ciliary body. And the ciliary body are basically these muscles that are able to contract and relax, and they will be able to manipulate the thickness of the lens, which will then be able to manipulate whether or not we focus on the far distance or the far sight or the near sight. And that would be done by the ciliary body that is pulling on these strings. And these strings are called suspensory ligaments. And we're going to discuss that in this lecture as well. Now let's talk about how the eye is able to focus on various light signals. And that's called accommodation. So we are going to discuss two types of focusing. One is the far focus, and the other is the close focus. So the way this ciliary muscle works is it's a little bit backwards compared to all the muscles that we would normally perceive. And that's because when the ciliary muscle is relaxed, which is going to be in the case of far sighting, so this ciliary muscle is relaxed. In this case, when it is relaxed, it's going to shrink. And when it shrinks, it puts a strain on these suspensory ligaments. So these suspensory ligaments are going to be elongated. And because they're elongated, now they're going to pull on the lens from both sides, and they're going to make the lens more flat. And because it is more flat, it is able to perceive light from a far distance. However, if we're looking at the contrast when we're looking close, so you're reading a book, or maybe you're looking at your phone, the ciliary muscle would be contracted. And as I've mentioned, they work the opposite of pretty much all the skeletal muscles that we would typically know as contracted, because contraction for the ciliary muscle means elongation. As you can see, this is going to be elongated, and therefore it's going to put this slack on the suspensory ligament, and it's not going to be pulling on the suspensory ligament. The strings here are not going to be pulling on the lens, and therefore the lens is going to be more round shape. So the suspensory ligament is going to be relaxed. Therefore, the lens is able to adjust by becoming more condensed and more round shape, and it's going to be able to adjust for a close accommodation. Next, we're going to discuss the function of the pupil. And the pupil's function is basically to allow light to enter the eye. And the wider it is, the more light it's going to let in. And of course, the tinier it is, the less light it's going to let in. So for those of you who are into photography or into filming or things to do with cameras, the way cameras work is you're going to have an aperture. So basically the same thing for the eye as the camera is, in terms of pupil adjustment, is the aperture, if you were to compare the two. And of course, if you increase the aperture, meaning that you're going to make the distance wider, or here you're going to have the diameter a lot wider, then you're going to capture more light and the image will be brighter. And if you condense it and you make the diameter very tiny, then you're going to be focused on a lot smaller spectrum. So you're going to have a lot less light coming in. And if you've noticed that typically when a person's outside and there's lots of light involved, then the pupil is not going to be dilated. The pupil is going to be constricted, and it will be very tiny because it's trying to avoid that so much light impulse coming in to the eye. In contrast, when you're under dim light conditions or at night when you're walking around, the pupil is going to be very dilated because it wants to take in all the light it can get. And so now we're going to take a look at how this mechanism functions, and it functions by reflex. So this is one of those reflexes that we test out as part of baseline testing when a patient comes in to the hospital, or if there's an emergency situation, then testing out the pupils is a good way to test out cranial nerves because we get to see the function of the reflex. So there's a couple of other things that affect pupil dilation or constriction, and that could be the distance of the accommodations, so the things that we looked at previously. So looking at objects from very far or very close while reading a book, or if you're looking at a plane flying very far, that is also going to have an impact on the dilation or the constriction, basically the diameter of the pupil. However, also stress and excitements or other chemical stimulants will also have an effect on the dilation of the pupil. And conversely, relaxation typically results in the pupil constriction. Now let's talk about the function of how these things actually occur. And the pupil contraction and dilation is controlled by two smooth muscles of the iris. And this is called the pupillary reflex. And of course, since it's a reflex, it's going to allow the pupil to constrict and dilate involuntarily, because this is, of course, done by the autonomic nervous system. We're first going to discuss constriction. So the nerve fibers from the oculomotor nerve signal the sphincter pupillae muscle, and it causes the constriction of the pupil. So basically this sphincter pupillae muscle is just going to cause constriction of the pupil. That's its only primary function. And this is done by the parasympathetic nervous system, because again, the pupil is typically constricted when the person is in a relaxed state. And for those who are wondering, the oculomotor nerve is one of the cranial nerves, one of the 10 cranial nerves that would be part that's coming off of the brain. Now, the other side of things, we're going to talk about dilation. And dilation is done by the dilator pupillae muscle. And this muscle is activated by the trigeminal nerve, which is another cranial nerve that's coming off of our brain. And of course, in contrast, this is going to be part of the sympathetic nervous system, because typically when a person is in fight or flight mode, they're going to want the most light coming in as they can so they can get a sense for the situation, and that means the person is very alert and is ready to go either to fight or to flight, run away. And so the way this works is the trigeminal nerve is going to activate the dilator pupillae muscle and through this sympathetic nervous system, and it's going to cause dilation. So the way this mechanism works is pretty simple. We have two different inputs. We have the parasympathetic and the sympathetic. Parasympathetic causes constriction because typically the person is going to be in a relaxed state. However, when the person needs more light to come in so they can assess the situation better and they can see things more clearly when they're in fight or flight mode, that would be activated by the sympathetic nervous system that will make the dilator pupillae muscle cause dilation of the pupil. So now we have our light coming in through the pupil and through the lens, and we have assessed those mechanisms. Next, it's going to direct the light to the retina and the rods and the cones. So let's assess the next part. We've talked about the light coming in through here. And finally, it's going to hit the rods and the cones in the back of the eye, which is going to be the retina. And typically, it's going to hit the focal point, where that is the part where everything is in focus. And it's not going to be your peripheral vision, but it's going to be the focused vision, which is where the focal point is at. So as you notice here, these ones are going to be the rods, and the cones are going to be these kind of colorful ones, the blue one here and those ones there. And what happens is they kind of going to hit this part that is reflective. So the retina is reflective, and it's going to reflect back, and the light is going to be caught by these cells. So these rods and cones are individual cells that are going to catch this light. Now, as you can see, the rods are much more numerous in number compared to the cones, and it is evident in this image. And what happens is then the information is collected from the rods and the cones in this part 3, and it's going to be connected together, and it's going to be relayed through the optic nerve that is going to exit the eye and go into the brain. So let's compare the rods and the cones and make a comparison of what they do and what they're used for and what their mechanisms are. So typically the rods are capable of scotopic vision, which is low light levels. So low light, we are going to be using rods. So the rods are going to capture that low light atmosphere. And because they have a low spatial acuity and do not mediate color, so there's going to be no color with rods. However, in comparison, the cones are capable of photopic vision, which is going to be higher light levels, so more light, and they're capable of detecting color vision, and hence they're also capable of high spatial acuity. So in terms of distinguishing color and the fine details of an image that we're looking at, that's going to be done by the cones. It is not going to be done by the rods. Typically, the rods are going to be responsible for that peripheral vision. As you can see, you're looking at this right now, your peripheral vision is not going to be as sharp. It's not going to be focused. It's out of focus, and you can kind of know the general thing that you're going to be seeing, but you don't have a great focus on those things. That's because the rods are the ones that are focused on that. However, the cones take in the light and they're able to perceive the things that you are exactly focused on. So the areas of fovea that is called, or the focal point, that is where the cones are typically going to reside in. There are three types of cones. There's the L cones, there's the M cones, and there's the S cones. And these are like sizes large, medium, small, but they typically depict different wavelengths. So the L cone is going to depict the long wavelength. The M cone is going to depict the middle wavelength, and the S cone is going to depict the short wavelength. And these are going to contain iodopsin pigment, and in contrast, the rods are going to take rhodopsin pigment. Now, the rhodopsin pigment is rich in vitamin A, and the importance of that is that it is responsible for night vision. Therefore, it is clear as to why the rods are sensitive to light. So rods are extremely sensitive to light. They're more sensitive to light than the cones because they're able to detect things at night. They're able to detect very low and very dim lights and create some sort of image out of it. So if you have a little bit of time, when you wake up and you wake up in the middle of the night, you need a little bit of time to adjust, and then you will be able to see things even though not much light is coming through. And that is because of this rhodopsin pigment that is able to, the way it works is once the light hits this rhodopsin pigment, it breaks up into two segments, and from there it is able to detect very low and very dim light. So that is the function of this rhodopsin pigment. The last thing we got to discuss is the optic chiasma. And the optic chiasma, we're going to have to label a lot of these things just to get a general understanding of how the optic nerve is now going to take the information down to the brain and how we're going to create an image inside our brain so we can understand what was captured, what kind of sensitive input was actually captured. So if we could evaluate this, we have the left field of view, or left vision field. I'm going to label it as LVF, and we've got the right vision field, RVF. Now the light is going to be coming through both sides of the eyes, and we are going to have several labels to go through here. So first we've got the optic nerve coming off from the back of the eye, and it's going to go through this optic chiasma. If you follow the colors here, the right field of view is going to hit this left part of the retina. Then the information from the left part of the right eye is going to travel and is going to intercross, and it's going to go to the left side of the brain. So this is the left brain, this is the right brain. And going to the left side of the brain, it's going to be perceived there. So from that, we can point out that the right field of view will be perceived by the left side of the brain, and the left field of view that's in red is going to be perceived by the right side of the brain. Basically, the input of the left field of view will be coming in from both eyes, the left eye and the right eye. However, both eyes will give information about the left field of view to the right side of the brain. And then both eyes will take in information from the right field of view, and all that information from the right field of view will be collected and given off to the left side of the brain. Now there's two other structures to label here. They're not as important to know, and this one is called LGN, which stands for lateral geniculate nucleus, and then finally this arrives at the primary visual cortex. Now in the primary visual cortex, our brain will be able to take the image from the right side of the brain and the left side of the brain and put them together in complete, kind of like in puzzle pieces, complete the image that you're seeing right now. Now that concludes today's lecture on the anatomy and physiology of the eye, and in the next lecture we are going to begin talking about the digestive system.